Chris Brady, and today I'll be teaching you about the hearing and balance structures. Let's go ahead and take a look at this model here. This is a model of the ear. The ear has three major areas. We have the external ear here, we have the middle ear right in here, and then we have the inner ear, this part right here. Let's start first with the external ear. So the external ear consists of the auricle or pinna, which is this outer shell-shaped portion, which is what most people think of when you say the word ear. Then we have the external acoustic meatus, or the external auditory canal here, that leads to what a layperson would call the eardrum, but what we'll learn to call the tympanic membrane. The external auditory canal is lined by skin all the way through, and the outer surface of the tympanic membrane is also lined by a thin layer of skin. And the external auditory canal, the skin that lines this, has lots of ceruminous glands that produce cerumen, or earwax. And that helps to keep your external auditory canal free of insects, and it helps to clear out dirt and other debris as it moves out. So sound waves are going to be collected by the auricle or pinna and funneled into the external auditory canal, and the external auditory canal will funnel those sound waves to the tympanic membrane. This is a model of the tympanic membrane. On the inner surface of the tympanic membrane, it's going to be attached to the auditory ossicles, specifically to the first one, malleus, and then malleus will be attached to incus, and incus will be attached to stapes, which is the stirrup-shaped one that is not shown here. When the sound waves are funneled through the external auditory canal, they start the tympanic membrane vibrating, and it transmits those vibrations to the auditory ossicles, which are connected to each other, and so they all will start vibrating, and that will push stapes into a part of the inner ear and create vibrations there. So we talked about the tympanic membrane and how when that starts vibrating, it will start the auditory ossicles vibrating. The auditory ossicles are located here in the middle ear. They're located in a space called the middle ear cavity, or the tympanic cavity. This is an air-filled space that's lined with mucous membrane. And there's a tube here called the eustachian tube, or the auditory tube, that connects the middle ear cavity to the pharynx, which is the upper part of your throat. This tube is normally closed, but when you swallow or yawn, it opens, and that allows it to equalize air pressure between the inside and the outside surfaces of the tympanic membrane. This is really important in order for you to have acute hearing, because the tympanic membrane can't vibrate properly if the air pressures are different on each side of it. Okay, so that's the function of the eustachian tube here. So this is the middle ear, and then I mentioned that stapes is going to be pushing in and out of part of the inner ear. So this structure here, this represents the inner ear. It's carved out of the petrous portion of the temporal bone. And there's an opening into the inner ear here called the oval window, and stapes will be pushing in and out of that oval window as it vibrates. We're going to take a closer look at the structures of the inner ear using another model. This is a model of the inner ear, and the inner ear is where all of the specific events that allow us to determine hearing and balance actually take place. So first, you'll notice this snail-shaped structure here. This whole snail-shaped structure is called the cochlea, and the cochlea is responsible for hearing, and we'll talk about how it does that in a little bit. We also have the inner portion here, which is called the vestibule, and it's got two membranous sacs within it, the utricle and the saccule, and these are going to be involved in balance. And then we have these loops called the semicircular canals. And then you'll notice that there are these bulges at the base of each of the semicircular canals. These bulges, each of these is an ampulla of the semicircular canal. And so these structures, the semicircular canals and their ampullae, are involved in equilibrium, balance and equilibrium. I'll also point out that we've got nerves coming in here to innervate these parts. This nerve here is the vestibular nerve. So it's innervating the structures of the vestibule right here, the utricle and saccule, as well as the semicircular canals. Okay, so that nerve, the vestibular nerve, this one is going to be carrying information about balance and equilibrium. I'm going to turn this model around so we can see the other side. On this side, we can see the other nerve that's innervating the inner ear here. This is the cochlear nerve that's innervating the cochlea, and so it's going to be carrying information about hearing. And these two nerves, the cochlear nerve and the vestibular nerve, will come together to form the vestibulocochlear nerve, cranial nerve 8. Now let's talk a little bit more about how we hear and the structures within the cochlea that will allow us to hear. So I'm going to take off part of this model and we'll look at what's inside the cochlea. So notice that this is part of the cochlea. So here we are looking inside the cochlea, and you can see the cochlear nerve coming in to innervate the cochlea. And notice that the cochlea is essentially, it's a spiral that continues around all the way up. And so we get repeating structures. So if we look in at one segment of that spiral, we will see 3 chambers. We have the scala vestibuli, the scala media, also called the cochlear duct, and the scala tympani. So here we have a larger model of the inside of the cochlea. So again, we have those 3 chambers, the scala vestibuli, the scala media, and the scala tympani. These are separated from each other by a couple of membranes. The scala vestibuli is separated from the scala media by this membrane here, the vestibular membrane. And then the scala media is separated from the scala tympani by this membrane here, the basilar membrane. We also have one more membrane, which is this one, and this is a pretty stiff kind of ridge. This is called the tectorial membrane. The tectorial membrane doesn't really move very much. The others are flexible and they can move. The tectorial membrane is less flexible. Another important structure here, and actually what is going to allow us to detect vibrations as sound, is this structure right here. This is the spiral organ of Corti. The spiral organ of Corti has these special hair cells that you can see right here. And these hair cells, when sound vibrations enter your ear, they make the tympanic membrane start to vibrate. That causes the auditory ossicles to vibrate, and stapes pushes in and out of the oval window. That starts the fluid that fills your inner ear, that starts that vibrating. And those vibrations travel through the scala vestibuli, and if they're within the range that we can hear, they will travel into the scala media and cause the basilar membrane here to vibrate up and down. And that will push these hair cells into the tectorial membrane. When the hair cells get pushed into the tectorial membrane, that causes them to bend, and they release neurotransmitter onto the cochlear nerve that is right underneath here, coming out along here. So this is the cochlear nerve coming along here. And so that sends the signals that we are hearing something. And the place in the cochlea, how far along the cochlea that it disturbs the basilar membrane, will help us to detect low sounds versus high sounds. So let me say that one more time. So remember that the cochlea is that long spiral. If sound vibrations disturb the basilar membrane early on, those are high frequency, high-pitched sounds. if they disturb the basilar membrane farther on, closer to the end of the spiral, the apex of the spiral, then those are lower sounds. And so that's what helps us to distinguish the pitch of the sounds that we're hearing. As those vibrations travel through the fluid in the scala vestibuli, fluid doesn't compress, so we need a release valve for that pressure so that it doesn't damage the structures of the inner ear. And so it's going to travel, those vibrations are going to travel all the way through the apex of the cochlea and then back through the scala tympani, and then they will push out through the round window, which is the second membrane-covered opening into the inner ear from the middle ear. And so the fluid will just push out into the middle ear cavity and then back, and that will release the pressure from those vibrations. So let's go back to this larger model of the inner ear. And so we've talked now about the cochlea and how hearing takes place in the cochlea. Let's talk a little bit more about the balance and equilibrium structures. So as I mentioned, within the vestibule, which is this whole area right here, we have these two membranous sacs, the utricle and the saccule. And each of these has a structure inside called a macula, which is essentially some hair cells embedded in a gel that has little tiny rocks, tiny otoliths on the membranes. And as you change your head position and your linear acceleration, so if you are accelerating in a car or if a car is stopping, then those little tiny rocks are going to shift around and cause shifts in the hair cells. And they will then release neurotransmitter on the vestibular nerve, on the axons of the neurons of the vestibular nerve. So these are the utricle and the saccule. And then the semicircular canals, inside each ampulla of a semicircular canal is another structure called a crista ampullaris. And that structure is going to detect rotational movement. So as you rotate your head, or as you do a spin on the dance floor, that's going to move the crista ampullaris structure, and there are other hair cells that will detect that movement and release neurotransmitter onto other neurons of the vestibular nerve. And one way that you can remember these structures, and which is the utricle and which is the saccule, is that these semicircular canals with their loops, they look kind of alien, like alien structures. And so you can think it's the US against the aliens, the utricle and saccule against the aliens. And so that's a fun way to remember those. Today I've taught you about the structures of the ear, those that are involved in hearing and those that are involved in balance and equilibrium. Now go ahead and practice these using the labeled and unlabeled images in your Learning Lab PowerPoints, and then you can apply these on the labs.